So before beginning, I would like to thank Dr. Nagla for her invitation, and I'd like also to thank the team of Faculty of Agriculture for their amazing organization and kind hospitality. Thank you very much. It's really gives me pleasure to stand here in front of my eminent professor and guest to talk about my, part of my work. Actually, this work entitled Structure Modeling and Docking of Viciacinera Cab8 Protein. This work has performed by my student Shaza Ahmed. She is teaching assistant and faculty of biotechnology, MSc University, under supervision of Dr. Rani Abu Ali, the head of nucleic acid and protein structure department at Agiri, and myself. Before my start, actually, in my work, I would like to expose why we do this work. Actually, we have, I've been in all over the world, big problem regarding the food. Actually, the food is not enough for all the people. This for several reasons, might be globalization, energy, there is food insufficient. I mean, actually regarding the FAO reporting, there is a reduction in a lot of crops like potato, wheat, maize, soybean, at least by 20%, which is really dangerous. According to their report also in 2010, they said that there is slight enhancement in the situation starting from 1990 till 210, but unfortunately still the situation is too bad. We have about 40% of people which is suffer from hunger. It's about 1 billion of people don't find their food, which is really big problem. Therefore, it's not surprisingly that all the people all over the world thinking about using biotechnology as a tool to enhance agriculture to solve the problem of food. Here in Egypt also, it is one of our opportunity. The Information and Decision Support Center put in their opportunity and suggestion to enhance our crops, to increase the using of biotechnology to solve this problem in Egypt. Actually, in Egypt, we have 7.8 million faddan cultivated. From them, there is about 2 million faddan are salt affected, which means we are losing our land. If you divide Egypt for the north coast region, for example, it really affected by salt concentration. Even the river itself, year after year, the salt concentration of the Nile River increase, which means we start losing our lands, and this will affect the availability of food in Egypt. Especially also after our revolution, that a lot of people start to building on our agricultural land. This means that we have, after a while, a big problem to find food. Therefore, we have a major goal. This major goal to enhance our crop. I mean to enhance the vertical and the horizontal ways to enhance our crops by increasing the yield or even by increasing the land which cultivated by good crops. To make a second approach, we need plants which could live in desert, which could survive under harsh conditions like drought, salt, and heat shock. Actually, I like the presentation it was today morning, which talk about that all these stresses regarding for water loss. I mean, all of them are regarding for water loss. So our problem, how our plants can live without water, it couldn't. But at least we could reduce the amount of water needed for these plants. So, our mission during this study is proteomic approach. Actually, this part from project running within our group. Our group thinking about having new gene, new engineered gene. This gene has special characteristics, like for example, having enhancing of function or even multiple function gene. This activity starts by isolating several genes. We success to isolate several genes related to salt stress and drought stress, and now we are thinking how can use these genes in proteomic approach to produce new candidate genes. Today I will talk about Cab8 gene. Cab8 gene actually work during oxidative stress. Reactive oxygen species elevated inside the cell during normal metabolism. Actually, it is really good for several physiological activity inside the cell. It can help the cell to survive during several conditions, but the problem if the reactive oxygen species increase above certain limit, it will start to order signal transduction for the cell to go to apoptosis. For that, it's really important to find some genes inside the cell to help the cell to survive under such a stress. Actually, oxidative stress considered as second stage after abiotic stresses. Cab8 gene is is a member of Cab8 family, which is a light harvesting complex found in thylakoid. It can capture energy and light, and at the same time it can help to survive under oxidative stress. The question is, as it's published everywhere, that Cab8 gene found beside the membrane or even inside the membrane. The question I would like to answer is Cab8 gene has a role in helping plant survive against salt and drought stress? Is Cab8 gene could help to replace water inside the membrane? This, from point of view of structure, we could at least put some indication of role of Cab8 gene. Cab8 gene was isolated by our group in 2009 from Viciacinera. So, first of all, we would like to investigate if Cab8 gene has any effect during salt stress or not. Secondly, we'll start to predict the primary, secondary, tertiary, quaternary structure of Cab8 protein. Then we'll start to determine the active site of the protein, and we start also to predict the active site and how it can combine, react with other protein. This study will help us to re-engineer the gene, re-engineer the protein, and then we could start to produce another kind of a super gene which could work more efficient or even have several functions. This sequence or gene related, it submit for database of Japan under accession number AB5862. The first step of our study was start to make phylogenetic tree to see how Cab8 gene is related from Vicia far away from other Cab8 family. We found that it's really the same clan with Cab8 gene isolated from Bisum stivum. Therefore, in our study, we will use the protein isolated from Bisum stivum as a model for us, as a reference. This is really important to know if we are going in the correct way or not. This is the first result of salinity test, just with bacteria transformed with Cab8 gene and controlled bacteria, and after different salt concentration, and we have enhancement in the salt tolerance of bacteria transformed with Cab8 gene. This is indicated by the optical density of the cells. Then we start to analyze the sequence, the amino acid sequence of the protein using the Whipple base of legumes. I mean, using the genetic code preferable for legumes, because the most Viciacinera is legume, and also it's really near to the Bisum stivum. After that, we start to analyze the domains to see the alpha helix, beta sheet, the hydrophobic and hydrophilic domains. It's really important to give us the quaternary structure of protein, which give us indication where is the active site and how it works. On the other hand, the hydrophobic part is really important, because if it is really have large part or large domains of hydrophobic like this, which is okay, so like this, and then this part could indicate, for example, that this protein located inside the membrane, and this might give us good indication that this protein work to help plant under salt stress or under water loss. Okay, thank you. After that, we start to make model system to see what's the quaternary structure of such a protein. Actually, we have 3 different models, model 1 and 2 and 3, and we use the protein which is isolated from Bisum stivum as a reference. Actually, model number 2 show about 93% homology for protein isolated from Viciacinera, while number 1 show about 83 and number 3 show about 34. From the beginning, we can say, okay, number 2 is best, which is wrong, because the modeling need to be really contain low amount of energy to be stable, enough bonds in the quaternary structure to be stable, and also to be functionally working. Therefore, we analyzed each model, and we found here that the best one is model number 1, which contain about it has score from stability and energy content about 1, and this indicates that the model number 1 is the nearest one for the reality. It's a bit far away from the Viciacinera, which is no problem, but it's stable, contain low level of energy content. After that, we start to analyze the number of alpha helix, or the parts or domains which make the alpha helix, and the turns. And we have here about 78 turns, and it's really important that this one indicate that this membrane this protein is inside the membrane and make turns outside and inside. We return back for our reference, which is the protein isolated from Viciacinera, and we make alignment according to regarding the motifs and domains we have found. And we have enough similarity to say that we have correct model which could make the further study on it. Here, just to test the stability and the quality of the structure, and we have nearly most of our residue above the threshold, which indicate that we have really good model of the protein. Here we start to investigate how many transmembrane domain we have according to the hyphobesity of this molecule, and we have 7 transmembrane domain, indicating really that Cab8 gene found in the thylakoid membrane. This means that Cab8 gene could work by somehow to help the thylakoid to stand under drought or salt stress, or even under losing water from the membrane. This is the structure of chlorophyll A. This is the most near protein to interact with Cab8 gene. So now question, our structure, our model has the ability to interact or to integrate with this protein or not? Regarding the analysis of the quaternary structure, yes. This is here. This here, the structure of chlorophyll A, and this our model, which is model number 1, and it's really a success in docking and can integrate together, which give indication that we have the correct model, and this model is inside the membrane and it could help in drought stress. So now we isolate Cab8 gene, and also in our lab we isolate several other genes like the hydrin. The hydrin is known as found inside the membrane, and then we could make a little bit change in the structure of both of gene to integrate them on one gene, and then we have new candidate gene which could work in drought stress, salt stress, and even in oxidative stress. We are sure that these proteins inside the membrane by the presence of 7 domains inside and outside, which are going inside to outside or outside to inside the membrane. The second step of our study, we will start to design a new model for the new protein, and we start also to test the real model in the lab. I mean by protein X-ray crystallography or NMR, to be sure that our model is correct, and we start to plan how we will make in the next model, inshallah.